Today, I want to kind of walk you through some of the things that we've learned about these plant-based herbicides, both in the field, orchards, and row crops, as well as in the greenhouse. Plant-derived natural herbicides usually fit into one of two categories. Either the active ingredients originate from essential oils or fatty acids. Essential oils are usually obtained from either pressing or steam distilling plant parts. Some of the original essential oil active ingredients registered for use in organic agriculture were clove and cinnamon oils. Both were steam distilled from their respective plants. One of the constituents of clove and cinnamon oil, eugenol, is used to reduce sprouting in stored potatoes by damaging bud sites on the tubers. D-limonene, a terpene found in the essential oil of cold-pressed citrus peels, is a next-generation plant-derived active ingredient registered for organic agriculture that is outperforming the older active ingredients. The second category, fatty acids, are composed of polar acid functional groups attached to non-polar carbon tails. This structure is similar to that of the plasma membrane and plays a role in their mode of action. Acetic acid, or horticultural vinegar, was one of the original fatty acid active ingredients registered for use in organic agriculture. Capric and caprylic acid are the next generation fatty acid active ingredients registered for use in organic agriculture, also outperforming their older counterparts. This graph here depicts the burn down or the efficacy of these treatments as a percent on the left, with 100% being total kill with 0 green plant matter. And at each different point of viewing these damage after the herbicide was applied. Throughout this presentation, capric and caprylic acid will be represented by CAP and all of the green in these graphs. D-limonene will be represented by LIM as all of the yellow in these graphs. Acetic acid or horticultural vinegar, ACET, will be gray. And cinnamon and clove oil, CIN, will be red. Again, both of these next generation active ingredients, both the essential oils and the fatty acids, outperform the original plant-derived active ingredients, with the burn down of the weed species increasing with time after application of herbicides, with the peak destruction occurring approximately 3 days after herbicide application. These next few slides will demonstrate what plots infested with Canada thistle and field bindweed look like 7 days after applying herbicides. First is the next generation fatty acid, capric and caprylic acid, with very few living plants in the tree row. Second is the next generation essential oil, D-limonene, with visibly dead field bindweed carpeting the plot in a reddish-brown coloration. Third, acetic acid, the original fatty acid active ingredient. Here you can notice there's visibly more thistle and field bindweed growth in this plot. Lastly, cinnamon clove oil, again the original essential oil active ingredients, showing more field bindweed growth, along with some field bindweed death in this red-brown colorization. These next-generation active ingredients outperformed their older counterpoints consistently throughout our study at multiple locations throughout multiple seasons. While next-generation plant-derived herbicides are highly effective at killing above-ground plant matter, they do not translocate to roots. This allows for perennial weeds and some annual grasses and biennials to grow back in about 2 weeks after herbicide applications. Each of these columns represent a treatment, starting at the far left with cap high being capric and caprylic acid at its highest labeled recommended dose, cap low being a lower concentration of capric and caprylic acid. Lim representing D-limonene, all compared to their synthetic counterpart glyphosate. As one of our farm partners who used glyphosate in a reduced tillage management plan, Cheryl Zachariason from the Zachariason Partnership stated, these burn down results are highly satisfying as they damage and kill plants quickly in comparison to many of the synthetic counterparts, especially glyphosate. This is a graphical representation of that burn down and regrowth of those Canada thistle plants we just saw, with percent cover on the left, showing the amount of green plant matter in each plot. And here with suppress and the green representing capric and caprylic acid, with the darker color representing its higher concentration and the lighter color its lower concentration. Yellow again is D-limonene, here with its trade name Avenger, and peak glyphosate. Although plant-derived active ingredients very quickly reduce living cover, perennial rhizomes create new shoots by day 14 after being sprayed with herbicides. Glyphosate, a synthetic herbicide that translocates systemically throughout the plants, can take up to 3 weeks to reach full efficacy. This slow dieback resulted in less Canada thistle growback, but no change was found in rhizome weight. Because of this growback, plant-derived herbicides were applied about every 2 weeks during the growing season throughout 4 consecutive seasons, in an effort to control perennial weeds Canada thistle and field bindweed. This graph shows early season weed pressure as a percent at the beginning of each of the growing seasons, with 2016 being a pilot year where only 3 applications were given. We found a decrease in early season weed pressure on plots treated with next generation active ingredients after a single season of multiple herbicide treatments. And this continued until that reduction in early season weed pressure was lost when plant-derived herbicides were applied less frequently during the remaining 2 growing seasons. This figure shows grass weed cover as a percent at the beginning of each of the treatments throughout each growing season. I want to highlight that the next generation active ingredient, D-limonene, was comparable in efficacy to hand weeding at reducing grass weed growth. This phenomenon was not seen with next generation active ingredients, capric and caprylic acid, showing in the green line. However, when we look at broadleaf weed cover throughout each season, both of the next generation active ingredients, D-limonene and capric and caprylic acid, are able to keep perennial weed pressure low compared to other treatments, including hand weeding, shown in the blue. However, these differences begin to disappear when plant-derived herbicides are applied less frequently, as we saw with the grasses. After seeing some of these differences in efficacy between plant species in the field, we decided to test next generation active ingredients on different species in a greenhouse setting. These plant species included both grass and broadleaf species of both weeds and crops, tested at multiple life stages with varying herbicide rates. Mayweed chamomile, stinking chamomile, or dog fennel is a difficult weed to kill due to its ability to grow back after killing above ground portions. This graph shows the Mayweed chamomile growback after treatments, with the green representing capric and caprylic acid treatments, with the lighter colors being a lower dose concentration, and the darker being the highest recommended concentration. The same is true for limonene. Purple here represents the control, and the hand-weeded plots completely killed all plants with no growback due to the destruction of that meristem. However, medium to high rates of capric and caprylic acid work better than D-limonene at reducing cover and growback at all life stages. Italian ryegrass was added to the study after farmer concern for weed biotypes becoming resistant to several of their herbicide mode of action, including lipid synthase inhibitors and amino acid synthesis inhibitors like glyphosate. Most of the next generation active ingredients were able to kill very young grasses. These charts will show the burn down as a percentage in blue above each of the treatments, with cap 3 representing the lowest concentration, cap 9 representing the highest concentration. Again, the same is true with limonene. And the growback represented in orange. Most of the next generation active ingredients were able to kill these young grasses, as this here shows. However, often when Italian ryegrass is of this size, we are unable to get machinery into the field, making control at this stage difficult. By the time the plants have grown to the 12-leaf stage, with leaves 6 to 10 inches long, only the highest labeled rate of capric and caprylic acid was able to kill all the Italian ryegrass plants, while D-limonene treatments killed all of the Italian ryegrass treatments, regardless of their concentration. Although no concentration of next generation active ingredients were able to kill all of the plants treated, once the plants reached the 34-leaf stage and the plants were a foot to 2 feet high, only D-limonene was able to completely kill individual plants with no growback at this stage. Another problem, annual grass and reduced tillage management systems, is rat tail fescue. Both next generation active ingredients work well to control this annual grass at medium to high concentrations, showing the difference in sensitivity between different grass species and the different plant-derived active ingredients. Because acidifying soils is a concern on the Palouse, many of our farm partners and interested parties have voiced concerns over adding acidic products to an already acidifying soil and its effects on the soil pH. These graphs show the soil acidity of greenhouse tested soils 4 different times, with green representing capric acrylic acid, yellow D-limonene, and purple the control. We did not find any acidifying of treated soil in both greenhouse studies or field samples. Here we have both the 0 to 2 inch depth as well as the 2 to 4 inch depths at a low organic matter site in Lewiston on the left and a high organic matter site on the Palouse in the right. The pH of the field soil was not affected at any of the depths or either of these sites. The mode of action of these plant-derived active ingredients is not shared with their synthetic counterparts, making them pride candidates to aid in combating resistant weed biotypes with their unique modes of action. So while these plant-derived herbicides cost more than their synthetic counterparts, they may play an important role in conventional agricultural weed management plans. Here we have some discussions that we would like to kind of talk about once we start to break into some groups. We're interested to kind of understand how some of these herbicides may fit into some of the conventional or other weed management plans. Even though they're expensive, would people consider using them to treat some of these resistant weeds? And kind of what are the technology setbacks or needs to increase implementation of spot treatments here on the Palouse?